Good afternoon, class 12 students of biology. We continue as we are coming to the end of chapter 14 of biotechnology and its applications. And in this chapter, children, if you remember, we had studied in this chapter in the last video, which was video 11, and in this we studied about biotechnology technique regarding crop improvement. We studied, children, about cell and tissue culture, where plants are developed from cell and tissue in aseptic laboratory conditions. We studied about some uses of tissue culture like micropropagation, somaclonal variation, and isolation of desirable mutants, etc. We studied about. Today we continue with the study of somatic hybrids. Now, what are somatic hybrids, children? Fusion of isolated plant protoplasts from two species can occur spontaneous, or we can induce it chemically, electrically, or by several other ways. Fusion of isolated plant protoplasts from two species, they can occur spontaneously, or we can induce the fusion of isolated plant protoplasts of two species by chemical, electrical, or several other ways. The protoplast fusion we see is a technique which offers a method for producing hybrids called somatic hybrids. So protoplast fusion technique offers a method for producing hybrids called somatic hybrids. And we see what is protoplast, children. If you remember, protoplast is the protoplasm of a living plant or bacterial cell whose cell wall has been removed. So what is inside is the protoplasm of the cell once its cell wall has been removed. So there are three potential uses of protoplast fusion technology. We see the production of somatic hybrids of genetically diverse species, transfer of one or a few chromosomes or genes between divergent species. The species evolved from the common ancestor are called divergent species. So we see that the protoplast fusion takes place between two divergent species also to produce a somatic hybrid. Then we see that production of cybrids is also seen. What are cybrids? Cybrids are cytoplasmic hybrids in eukaryotic cell produced by fusion of whole cell with a cytoplast. What are cytoplasts? They are enucleated cell. So we take a cytoplast and we fuse it with a eukaryotic cell. And when we fuse the cytoplast with eukaryotic cell, then we see that fusion of the cytoplast with eukaryotic cell, it produces a hybrid. This combined hybrid is known as a cybrid. And we see that production of cybrids for transfer of organelles such as nuclei or mitochondria and chloroplasts from one species to another is done by the making of cybrids. Then we see that there are interspecific somatic hybrids which have been produced in a number of genera. which includes Brassica campestris and the Datura plant, in Oryza, in Petunia, the flower that you have seen in our school, the Petunia flower, and in Solanum. We can produce somatic hybrids. Then we see that these hybrids thus produced, they showed new gene combinations which can be utilized for conventional breeding programs. We see that hybrids that were produced, they had many new gene combinations, and so these hybrids can be utilized for conventional breeding programs. The somatic hybrids have been produced between distantly related species which are distantly related. So from those species, the somatic hybrids have been produced, children. So we see that another exciting application of protoplast fusion technology involves the transfer of genetic information from the cytoplasm, which is carrying the genes for traits like hybrid vigor, the strength of a hybrid plant to produce in great numbers. Disease resistance is another genetic information that is present when we apply the protoplasts or we apply the genes. Then there are certain protoplast fusion techniques which give herbicide resistance and cytoplasmic male sterility. So this is located in the plastids and the mitochondria. So these kind of resistances, they are located, male sterility, these kinds of cytoplasmic resistances are located in the plastids and mitochondria. What is male sterility, children? That in this, the male portion of the plant, it does not take part in reproduction. So that is why they are male sterile areas of the plant. Then we come to microorganisms like nitrogen fixing bacteria and cyanobacteria into protoplasts have been incorporated. And when we incorporate the microorganisms like nitrogen fixing bacteria and cyanobacteria, this has been carried out for establishing endosymbiotic association of prokaryotes with cells of higher plants, which means that there is a symbiotic association between prokaryotes and higher plants. For example, the higher plants, they give nourishment to the prokaryotic cells, and in the bargain, the prokaryotic cells, they fix the nitrogen, and by putting this trait of nitrogen fixation into the protoplasts, we see that non-legume crop species are developed. Those crop species which are not leguminous but still they are able to fix nitrogen. Then we see hybridization through embryo rescue. We see that hybridization through embryo rescue technique has been followed. In embryo rescue technique, this allows wild varieties to cross with the cultivators or the cultivated plants. Here we see that the wild variety, they are cultivated they are crossed with the cultivated plants. So when this happens, that the wild variety is crossed with the cultivated plants, then we see that the varieties are born which have got greater resistance to pests and diseases and they produce better quality grain. So that is why hybridization through embryo rescue is done. This technique helps to improve groundnut, pigeon pea and chickpea. All these varieties of plants are improved for better yield. Semi-Arid Tropics Institute, Hyderabad, known as ICRISAT, has developed a new hybrid pigeon pea, and this variety is known as ICPH8. This is a pigeon pea variety that has been developed in Hyderabad Semi-Arid Tropics Institute, and this matures in 100 days. In place of 200 days, this variety can mature just in 200 days, and it is a much more yielding variety which yields 25% more than the normal variety yields. So we see that the above technique, the embryo rescue technique, has been developed in genera like Triticum, in Avena, in Oryza, in Brassica, causing improvement in crop plants, and many wheat varieties have been transferred from rye. We see barley successfully crossed with rice, maize, wheat and rye give very strong disease resistant variety plants. Then we see that anther and pollen culture technique is being used. It's a culture by which haploid plants can be obtained from anthers or from microspores. So we see that this is a new technique in which the anthers, which are of course haploid, they can be used to make full plants, or microspores from the microspore mother cells, which are the pollen, they can be utilized for bringing up haploid plants. After the success of using this technique in Datura by Guha and Maheshwari in 1964 to 67, this technique of anther culture has been utilized for production of haploids in wheat, in barley, maize, rice, rye, in triticale, in brassicas, in potato, in tobacco, etc. If desired, diploid plants can be easily produced by the haploids also. If we want to change the haploid plants, children, into the diploid plants, which use the fusion of cells both of the female and the male, then we can utilize a plant product that is found inside the plants known as colchicine. And that colchicine treatment, it changes the haploid plants into diploid plants. This treatment is the treatment of using medicines derived from colchicine found in plants. Then we see that some methods of obtaining haploids in crop species include anther culture, culture of unpollinated ovules. Then we can use culture of isolated pollen, and embryo sacs even are utilized for bringing up haploid crop species. Use of chromosome elimination is there. The above method is called as Bulbosum Method, and this technique has been used in barley. For example, if you see here, this has been used in barley, this technique, and this barley plant has been given the scientific name Hordeum bulbosum. And this technique of growing haploid plants has been used in barley. The Chinese, they have produced almost 80 varieties. Here we see is the Bulbosum method, which has been used for barley. Then we see the Chinese, they have produced 80 varieties of rice by anther culture method, Zinzua, Hua U1. This method has been utilized by the Chinese for causing about 80 varieties. In wheat also 20 varieties have been developed, like Yunhua 1 and Yunhua 2 varieties have been developed. And these varieties are rust resistant, cold resistant. These varieties of plants are very high yielding plants, and they are early maturing plants. Like we saw that plants can be produced just in 100 days instead of 200 days when we talked about the hybrid pigeon pea variety. So we see that early maturing plants are obtained by anther and pollen culture. Then we see this anther culture technique was used for improvement of sugarcane with tall stem and higher sugar content. The sugarcane which were made by the anther culture technique showed so much improvement that the stems were fatter, taller, and more sugar producing. Then we see haploid barley are resistant to mildew, to barley yellow mosaic virus, etc. So haploid barley is much more stronger, much more disease resistant, and high yielding variety which produces much more food to feed the millions in our country. So children, after this we will study about some important applications of biotechnology in agriculture, especially in the field of pest resistant plants, in the next video. So till then, wish you all the best. Have a good day. Thank you.